Today, we're going to pick up on protein structure, and we're going to be talking about one of my all-time favorite topics, which is how do you go from a sequence of amino acids to this truly beautiful structure right here. This is actually a good friend of mine that I spent 5 years writing my Ph.D. about and studying and doing experiments with. So this is truly one of my all-time favorite topics in the 10 weeks that we talked together. Okay, so that's our goal. Understand how proteins work, understand what their composition is, and then we'll get into conformational analysis, amino acid conformations, and then finally looking at protein structure. So this week is all protein structure. Next week will be all protein function in our scheme. Okay, any questions about where we're going? Okay, a couple quick announcements. Read Chapter 5, work the odd problems, as usual. There'll be a couple of topics that I'll have you skim. The synthesis of peptides, for example, we're just going to skim through. We just don't have enough time to talk about it in great detail, but it's in the book if you want to read more about it. Book reports, sorry, this should be journal article reports, are due this Thursday, Valentine's Day. A week after that, the abstracts are due for your proposals. Okay, so you should probably be thinking about your proposal topics now and starting to do some research on them and coming up with an abstract. On Thursday, I'll give you the format for this abstract. Okay, but for now, don't panic about this. This is a single paragraph that you're going to write describing your proposal idea. Okay, and this will be worth 10% of your proposal grade. So it's actually a really important part. This gives me a chance to give you feedback, and I'll give you a format that it's basically a formula that you could follow. Be 5 or 6 sentences, pretty straightforward. But in order to do it, you do need to have your topic. And it's almost impossible to think of a good topic the night before it's due. So you should be thinking about the topic now, okay? Coming up with a proposal topic this week. And again, this journal article report is designed to help guide you towards coming up with your own idea as well. Okay, and I'm also going to ask you today to learn the 20 amino acids, their structures, 1-letter and 3-letter codes. But I'll have more to say about that shortly. Okay, office hours. Jeez. Okay, office hours are going to be today. And let's see. Office hours today by Krithika, and then office hours tomorrow by me, and then office hours Thursday by me again, and then office hour Friday by Miriam. Okay. Any questions about office hours stuff? Okay, things like that. Okay, so one question that I get asked a lot during office hours, it actually came up last week, but it comes up every year, is something along the lines of, I have a vague idea that I want to do something in medicine, but I'm not sure exactly what that is. And so I want to take just a moment to sort of talk to you about careers in chemical biology and careers in the health profession, okay, professions broadly. Okay, because I think this is something that we don't spend enough time talking about when it's where 99% of you are going to go after you graduate. Okay, so all of us, I think everyone in this room has one goal, which is to treat people. Okay, at the end of the day, I think all of us want to make the universe a better place. We want to make people healthier and happier. And so our overarching goal, I would say, of everyone in this room, I suspect, is something like this. So in the end, we want to have people who are happy. Okay, that's our ultimate goal, right? And I suppose if anyone's really interested in making money as a way of getting to this, then you're probably in the wrong class. You should be taking a class in real estate or I don't know, copyright law or something like that. But there's other classes on campus that can get you to the dollar signs. Okay, so I want to talk to you today about all the different ways that you can contribute towards treating patients. Okay, which is I think the goal of most of us in this room. Okay, so the most obvious way is to treat patients by being a medical doctor, an MD. And I think this is what many of you have on your minds. Of course, MDs come up with will prescribe pharmaceuticals, which are then given to the patients by pharmacists. And again, I think that this is probably the goals of, so these are all contributing in here. And then, of course, there's optometrists. So there's DOs, which are also participating. There's, there's just other types of, there's dentists. All of these are sort of the hands-on ways that, that we think of when we think about treating patients over here. Okay, but what I want to talk to you about are all the other ways that you can contribute to this ultimate goal that don't involve, say, going to medical school, that don't involve, becoming a dentist or something like that. I think you're all aware of what it means to become a dentist and, what it means to become an optometrist, okay? But ultimately, someone has to come up with the drugs, the pharmaceuticals, the therapeutics that the pharmacists are going to put in. Pharmacists are going to put in bottles, and that MDs are going to prescribe. And so before you can get to this stage over here, there's a whole series of clinical trials that take place. Okay, so the clinical trials are staged into 4 stages, where the first stage is looking at toxicity, and the 4th stage is looking at dosage, and every stage is looking at efficacy, which is to say effectiveness. Okay, so someone has to do those clinical trials, and the people doing these clinical trials these clinical trials include scientists, so PhDs, but also MDs are running these as well. MDs are thinking about what dosage to use, and pharmacologists play a role as well in designing these trials and looking at side effects and interpreting data. Okay, now on the PhD side of things, there's PhDs who are analyzing data, who are thinking about side effects, who are thinking about all kinds of aspects of the trial. They're thinking about the data that, say, DNA sequencing data. If it's a trial that involves a particular mutation of a cancer-associated protein, someone has to be thinking about the underlying biology, the underlying chemistry involved. And so this is one role that students from my laboratory play. So I have students in my laboratory who design clinical trials, graduated from my lab, who have PhDs in chemical biology and are now designing clinical trials and interpreting the data. Okay, so now in order to get the drug to the clinical trials, it turns out it's a long process. It takes about 10 years or so to get a drug into a clinical trial. And so kind of at the very beginning of all of this, a series of experiments are done over here by biologists and chemical biologists. Okay, and the goal here is to identify potential targets that would be useful in organisms, in humans. Okay, and it might not start with humans. It might start with, say, I don't know, zebrafish, where maybe you identify a particular mutation that causes heart muscle to grow back in zebrafish. And zebrafish are this clear organism that you can actually look in and see their hearts. Okay, so dude, if you find something else, find somewhere else to go. Okay. Okay, so someone has to do that. Someone has to get the foot in the door and identify what the ultimate target is going to be, that this cancer thing is going to target. Okay, so this again is done by biologists and chemical biologists, and these are at all levels. So you tomorrow can go out and when you graduate from UCI, you can participate in this at your level of a Bachelor of Science. You can have a master's, you can have a PhD. All of these give you a way of identifying targets. Okay, so we'll call this ID targets. Okay. Now, once the target is in hand, then someone has to chemically synthesize the drugs, right? Someone has to synthesize the initial leads that are then going to eventually become the therapeutics. Okay. And so this is typically done by medicinal chemists. Okay, so medicinal chemists but also this is done by chemical biologists who devise assays looking at how effective the compounds are. Computational chemists play a key role. So someone has to model whether or not the pharmaceutical are actually targeting the drugs as expected. And in doing this, they can start to decide whether or not they're getting more effective compounds. They can use this to design compounds that bind more effectively to the target and avoid side effects. So this, again, is done by people who have BS degrees, MS degrees, and PhDs. OK, so let me just talk very briefly about this. So if you went out tomorrow and got a job at, say, Genentech as a BS chemist, let's just say BS chemical biologist, and you worked in Department of Protein Engineering at Genentech, what you would be doing is you'd be working as part of a team to synthesize compounds, perhaps if you're working in a medicinal chemistry team, or if you're in, say, protein engineering, you'd be synthesizing proteins, you'd be testing proteins, you'd be trying to gauge the efficacy of those things and trying to decide whether or not things are working. Okay, and your role on this team is you're kind of like the foot soldiers. Okay, you're the muscle that makes it possible to do the science. Okay, now the thing is, if you're working at a really good company and you're doing amazing science, eventually you can get promoted up the ranks to be at the same level as a PhD scientist. Okay, so when I was at Genentech, I had a friend who never got a PhD, but he published 7 papers in science when he was there, and they took that as the equivalent of a PhD, and he was promoted to the levels of scientists, which are usually reserved for people who have PhDs. The PhDs are running the teams. They're typically running teams of about 4 people, and the people on those teams include a couple of BS level people, maybe 1 master's. The master's person is kind of like a super technician who's kind of corralling the resources, directing things, helping things along, pushing things more quickly, that type of thing. Sometimes they're called research associates, but it's the same sort of idea. Okay, so in here, so there's lots of opportunities in here, and then eventually someone has to do things like test the compounds in animal models, and then also synthesize enough of the compounds before you can get to clinical trials. And so actually, I'm missing an important step over here, and I think what I'll do is I'll have this. So we'll call this scale up. Okay, someone has to scale up the compounds. And this scale up is typically done by process chemists. Okay, so if you're that person who loves organic chemistry and loves mechanisms, this is for you. Okay, you're going to be basically really understanding the details of a particular reaction and trying to improve its yield so that you can get 10 kilograms of this stuff, enough to run a 1,000-person trial or something like that. Okay, so someone has to do this, and again, this includes some BS level people, but also MS and also PhD. I haven't talked very much about the MS. MS is a good thing if you don't plan to go on to sort of a leadership role on the team. If you're totally happy at kind of an intermediate level, a master's of science gives you a higher salary. And over the course of a 30-year career, a difference of $10,000 a year comes out to a lot of money. Okay, especially with compounding. Okay, and so this gives you just a little bit more freedom, but not a lot of freedom. Really, the most sort of creativity and freedom is at the level of PhD until you earn it if you're at this BS MS level through just being successful. Okay, so anyway, in the end, this leads then to the compounds, which then can be tested in the clinical trials, which ultimately leads over here to the happy patients. Okay, so a couple of thoughts about this. So I have a diversity of students in this classroom. I have one guy over there who text messages through every class. It drives me crazy. Doesn't even seem to listen to me at all. But I also have some people who have 4.0 GPAs. Okay, so I have a wide range of students in a class like this. And I have a message for everyone. No matter what your GPA is, no matter how well you're doing here at UC Irvine, no matter if you're text messaging through every class here at UC Irvine, there's a place for you in this majorly challenging enterprise. To beat a disease like cancer, we need everyone. Okay, it's not true that just the 4.0s get to go on and go on eventually to win Nobel Prizes. There is a role for everybody. And I've seen people with 2-3 GPAs coming out of college who have gone on to win presidential awards for their quality of their science. Okay, and that's one of the cool things about science, is it's a total merit-driven enterprise. So if you are really good and you work really hard and you bring a lot of heart and you put away your cell phone at key moments, you too have a role for this. Okay, and so let me talk to you very briefly if you want to be over here and your GPA doesn't let you do that. Okay, so first, if you're applying to medical school, you should be thinking about having like a 3.6 or higher GPA. I've had trouble getting students into medical schools with 3.8s. It's that unpredictable. Okay, MD, PhD, you should be having like a 3.95 GPA. Okay, and like Ph.D. graduate school, you should probably have somewhere in the order of 3.3 to 3.5 minimum. Okay, now don't panic. Okay, if your GPA is below that, there is a place for you, and I want to talk to you very briefly about how to get there. Okay, so number one is you can start at these BS levels positions in a company. And as years go by, your GPA from your undergraduate becomes increasingly less important. If you're at this company and you're doing really good science and you're impressing people, then you know that calculus class that you got a C- in that's been bogging down your career ever since suddenly becomes a lot less important. Okay, so every year that goes by, your GPA gets less important. Okay, and then at that point, then it's all about heart and your ability to rise to the occasion. Okay, which is good news. Now the other thing is, if you want to go straight towards a PhD and you don't have, say, you know, a 3.6 or 3.8 GPA, don't panic either. What you could do is you can go off and get a master's degree at a Cal State and then come back to get a PhD at one of the top universities in the country. I would suggest that you don't go down to a low-quality institution for getting a PhD. Okay? This is not elitism. Okay? You can look up my own record. Obviously, I have a lot invested in high-quality universities. But the point is that we actually have something like 200 chemistry PhD granting universities in the United States. Of those, most of the employment for the kind of top-level jobs in both industry and academia are coming out of probably 50 to 60 of those. Okay, so if you drop down to, say, you know, a third-tier or fourth-tier PhD, you can get a PhD, but it's going to be hard then for you to come back. So a better way to do it would be get a master's degree. Cal State Fullerton, Cal State LA are outstanding institutions. You get your master's from there, and you show that despite, you know, that bad grade in calculus, you still are someone that we should take a bet on. And then you come back and get a PhD at, I don't know, UC Berkeley or UCLA or Harvard or Johns Hopkins or any of the great institutions here in the United States. Or maybe you go abroad and get a PhD from Cambridge or you know, one of the good schools in Australia or so on and so forth. Okay? So there is a role for you at whatever level you want to participate in this major enterprise of treating patients. And the real question that I want to challenge you with is, what do you want to do? Are you the kind of person who likes to think about molecules at the atoms and bonds level? In which case, then you probably want to be somewhere over here. If you're the kind of person who likes customer service and likes, you know, having patients coughing on you, and you know, you don't mind blood and stuff like that, you probably want to be over here. Okay, so you basically get to dial in whatever level of interest is ultimately yours. Okay, and no matter what your current GPA is, there is some room for you. Okay. Any questions about that? Okay, so again, this comes up every year during my office hours, and this year I thought I'd make it more systematic and just tell everyone. Okay, no questions? Okay, oh, you know, one last thought. None of this is set in stone. You know, the great thing about United States is that you can reinvent yourself. So if you end up going to dentistry school and you don't like it, change. Okay, I've had friends, actually I have a friend who got a master's in computer science and was a programmer, and he hated it. He absolutely hated it. So he went back and got a degree in dentistry, and he's now a really happy dentist. Okay, so no matter what you're doing, you don't necessarily have to get locked into one thing. There's considerable room to reinvent yourself and then use that earlier experience to be more successful at your next career. Okay, so that's one of the great things about this, and that's one of the things I really prize about our system. Okay, any questions? Okay, well, let's stop here. I want to move on, and I want to talk about proteins next. Specifically, I want to talk to you today about protein structure. Before I do, I need to review what we talked about last time. So last time I was telling you about RNA and how RNA had a diversity of different structures. What we saw last time was mechanisms of RNA polymerase. We saw this last week. But our message on Thursday that was after the synthesis of the mRNA, heavy modification takes place. We saw that both ends of the mRNA are capped. They're altered. There's splicing events. There's cutting and pasting that takes place after the messenger RNA is synthesized. For that matter, after synthesis of RNA, there are other covalent modifications that are taking place on the RNA. And we saw an example of that being transfer RNA, tRNAs, that are synthesized, that have modifications to their bases. Those modifications, as we saw, have important functional consequences in terms of determining the specificity of the amino acid that's appended to the 3' end of the tRNA. Okay, we talked a little bit about that and how amino-acyl tRNA synthetase reads those types of modifications out. It reads out the anticodon, and in the end, the correct amino acid is appended to the correct tRNA. RNA, as we saw, is also a very versatile biopolymer beyond its ability to be both an mRNA and also a tRNA. For example, we saw catalysis by RNA, and this was catalysis by a class of RNA-based catalysts that we now call ribozymes. And it turns out this actually works very, very well. We also talked a little bit about binding activity from RNAs by aptamers. And so all these things make RNA this really versatile molecule that is really ripe ground for all kinds of discoveries. Whenever I pick up Science or Nature, there's something new about RNA in there. And the chapter that I had you read has large sections of it that are changing, that are being transformed overnight by discoveries that are being made in laboratories here at UC Irvine and here at other universities across the world. So this is a really exciting area to investigate. It's an area that I really encourage you to think more about because RNA is so versatile that it continually surprises us. Okay, let's talk about proteins. So we're continuing our journey down the central dogma of molecular biology. We've talked about DNA, we've talked about RNA, we're now at proteins. And proteins are named proteins because they were thought originally to be of central importance to the cell. They still are of central importance, despite everything I've told you about of RNA and DNA. And I'm showing one of my own little biases. But here's a great picture of Max Perutz on the left and John Kendrew on the right, who shared the Nobel Prize in 1962 for determining the first three-dimensional structures of proteins. And back in the early days of the structural biology, they would build little wireframe models of proteins. This is before computational modeling was available. And so there was actually tremendous craft involved with building protein structures. And there was almost this tactile feel as you'd have these things in your hands. We still have a little bit of that in structural biology. Nowadays, we use 3D printing, but it's the same idea. And so we like to think about things in 3 dimensions. And one of the things that's always entranced me about this topic is that we're going to be taking things off of the flatland and talking about them in 3 dimensions. Okay, so the first thing I'd like to ask you to do is memorize the structures of the 20 amino acids and their abbreviations, both the 3-letter and the 1-letter codes. Here I'm depicting just the 3-letter codes, or just the 1-letter codes. And the amino acids can be roughly grouped according to the functionalities of their side chains. So at the very top, here are some side chains that are hydrophilic, including acidic functionalities like aspartic acid, which has abbreviation D, glutamic acid, abbreviation E. but also the hydrogen bond donor, tyrosine. Here are some aromatic amino acids with aromatic side chains, and then some basic side chains. Please do this as soon as possible. Starting on Thursday, I'm just going to assume that, that I could talk about these without kind of referring back to, ooh, histidine has this imidazole functionality, tryptophan has this indole functionality. So start memorizing this right away, okay? That will really help your understanding of what's going on. In addition, there are other functionalities that are fully hydrophobic. These are the aliphatic aliphatic amino acids shown here. Okay, and then the last of our 20, there are some that contain sulfur. This includes methionine and cysteine. And then some other ones that are hydrogen bonding polar amino acids. Some of these, 2 of these in fact, have a second stereocenter at the beta carbon. Notice that we're going to be calling the carbon that's alpha to the carbonyl. We're going to call that the alpha carbon. And then if you go alpha to beta, the second carbon away is called the beta carbon. And threonine has a second stereocenter at this beta carbon. I didn't point it out, but the other amino acid that has a second stereocenter at its beta carbon is isoleucine. Okay, so isoleucine has an S stereocenter in the Kahn-Prelog-Ingold rules for assigning S and R stereochemistry. Threonine has an R stereocenter in that position. Those 2 you just have to memorize. There's no way around it. Okay, but good news, for the other 19 of the 20 amino acids, this stereocenter at the alpha carbon is S. Okay, so every single one of these has an S stereocenter at this alpha carbon. The one exception is glycine, which does not have a stereocenter at its alpha carbon. Okay, makes sense? Okay, I expect that you might have seen this in Bio 99 or something like that. If not, don't panic. Just go ahead and memorize these as quickly as possible. Okay, any questions? All right, let's move on. Okay, so when we refer to the amino acids, what our convention in this class and the convention in biochemistry and chem bio is that we're going to refer to them using the one-letter codes followed by a number. That number designates the position of this residue. Residue is another way of saying amino acid within the larger protein. Okay, so if you had a protein that has 180 amino acids in it, and at position 171, the amino acid was a histidine, you would designate this as H171. Okay, and so on. And obviously, this is a manganese ion, and so it's just going to get the regular elemental designation. Okay, so anyway, that's the convention we're going to follow. Pretty straightforward. Okay, so here's what I want to do over the next couple of days. I want to understand the forces that take a set of amino acids, like what I showed on the previous 2 slides, or 2 slides back, and then understand how these amino acids force the protein to adopt a particular shape. Okay, because the sequence of amino acids that encodes a protein in turn specifies one predominant shape. Okay, and this is actually kind of a pretty wild concept if you stop and think about it. But again, if you have a particular sequence of amino acid, that in turn will dictate one particular shape. Okay, there are some proteins that refuse to have any particular shape. They're largely disordered. Those are kind of the special exceptions. So for this class, where we're kind of at a foundational level, we're only going to be talking about proteins that adopt one predominant shape. And if you start to look at things more closely, there are a lot of exceptions, but we're not going to worry about that. Okay, so again, our goal is to understand how you go from this to something like this. Okay. Understand the forces behind protein structure. Okay, so the first thing we have to talk about is what happens when you string together the amino acids. So first, the backbone of the peptide or protein has some directionality associated with it. And oh, by the way, I'm going to use the words peptide and protein interchangeably, but they're not. Okay, so peptides are short sequences of amino acids that are connected together by amide bonds. And in turn, these are so short that they don't adopt any one fold or one particular structure. Proteins are long enough so that the amino acids force the peptide biopolymer into a single conformation. Okay, so it takes a certain minimum length. Short peptides that have, say, 20 amino acids or less are typically so largely disordered. When we put them in an NMR tube, we see all kinds of things. Okay, there's all kinds of different conformations, and these things are just kind of flying all over the place. If we make the protein longer, if we add amino acids on, oftentimes we can get to something that's more structured. Okay, at least, especially if we're borrowing from sequences that are known to be structured. Okay, so now we have to talk about an important convention in designating these sequences of peptides and proteins. We're always going to list the sequences in order from N-terminus on the left to C-terminus on the right. Notice that the C-terminus is the carboxyl end of the amino acid. So in each case of these amino acids over here, we're going to have the carbonyl or the carboxylate on the right and the amine on the left. Okay, and something that's really important is the these sequences that go from N to C cannot be reversed without resulting in a different peptide sequence. The two sequences, the one on the top and the one on the bottom, are constitutional isomers of each other. They're not diastereomers. They're nothing more than just isomers. And the way you can tell is notice that the positions of the carbonyls and the NHs are all wrong between these two. Okay, in fact, it's very likely that these two will have different binding profiles. It's very likely that these two are going to have, say, different flavors if we were looking at a peptide that had a sweet taste, for example, because the arrangement of the carbonyls, the arrangement of the NHs will really determine hydrogen bonding to the backbone of the peptide. And typically, say a taste receptor, if we're talking about sweetness, a taste receptor will have hydrogen bond donors and acceptors that will interrogate that backbone and want a certain arrangement of that backbone. And the wrong isomer is likely not to bind to the target. I'm using the word likely because molecular recognition is very hard to predict, but it's very likely that it's not going to bind. Okay, so these two molecules are different, and that's why we have to have the convention that we're always going to write these with the N-terminus on the left and the C-terminus on the right. Notice here that I am not listing the N-terminus and the C-terminus. It's kind of like when we talked about DNA sequences. We had a 5' end and a 3' end, but oftentimes we didn't list the 5' or the 3', right? We just assumed that they were there. So similarly with amino acids, sometimes people write H2N on this side and CO2H on this side, but most of the time actually people just leave it out. Okay, it's kind of understood. N-terminus is always on the left, C-terminus is always on the right. Okay, okay, so that's the convention that we're going to follow. Here's something else that's extremely useful for you to know, to the point where I'm kind of tempted to ask you to memorize it, which are these pKa's. I haven't asked you to memorize a lot of pKa's if you took 51C with me or if you took earlier courses with me, but these actually are very useful. And the pKa's that you need to memorize are all shown here, and you don't have to memorize them to the nearest decimal place. Just round them up. Okay, so just come up with something that's roughly equivalent. So what I mean by that is just know that these carboxylate side chains will have pKa's on the order of 4 to 5 or so, somewhere in that range. And the reason why the exact numbers are not so important is that the environment that the functionality finds itself in can dramatically alter the pKa. And this is a property that proteins are notorious for taking advantage of. You can have, for example, a carboxylic acid side chain that's positioned in a way that makes it much more acidic than the pKa that's written behind me might suggest. Okay, so proteins will tailor the environment that these functional groups are going to be found in, and in doing so, drastically alter at times the pKs of the functionalities. So I want you to have just the range of the pKs in mind. You should know, for example, that when a peptide like this one is dunked in pH 7 water, which is obviously the pH 7 being neutral, here's what we would expect. Okay, we'd expect to see these carboxylates deprotonated. We'd expect this tyrosine to be protonated. It's only 0.313% deprotonated at this pH, etc. Okay, so the C terminus is going to be deprotonated. The N terminus will be protonated at this pH. Okay, and geez, this, I realized I wrote this last year, but it's kind of true. Okay, when I think about proteins, first thing that comes to mind are charge functionalities. Charge functionalities control a lot of the ability of the protein to catalyze things, to bind to different things. So the first thing that you absolutely need to know is what is the charge? Okay, and you kind of need to be able to predict that quickly. Okay, so I'd like you again to memorize these pKs, just the range. Okay, not exact numbers, but just the range. Okay, so these side chains are going to dictate, as the side chains interact with each other, that in turn is going to dictate the folding of the protein. Okay, so this goes back to the major challenge that I set up earlier. How do we go from amino acids to this complicated three-dimensional structure? What we find is that the side chains are going to be packing against each other, and that in turn is going to largely dictate particular folds of a protein. Okay, and evidence of this can be found in the fact that the trends in side-chain interactions mirror the strengths of these interactions. Okay, so the side-chain interactions that are found most frequently are also the ones that are the strongest. Okay, so for example, at the very top over here, we have phenylalanine binding to phenylalanine. These are two amino acids that have benzyl functionalities. These benzyl functionalities, large aromatic phenyl groups, can pack against each other quite readily. And so for this reason, this side chain is highly enriched when we look at sort of statistical analysis of side chains found in structures. Okay, so this is a very strong interaction, and it's therefore used quite a bit by proteins to set their structures. On the other hand, the weak interactions, this is one that involves the amino acid glycine. These are very rare. Okay, so glycine doesn't have a side chain. Glycine is the one amino acid that had no stereocenter. It has two hydrogens at its alpha carbon. And this one does not form good interactions with the other side chains. And so for this reason, its interactions with other amino acids, very rare, and its interactions very weak. Okay, make sense? Okay. So let's talk a little bit about the specifics of those interactions from one side chain to another. What we find when we look at this is that the strongest ones are most common. That's the point of the previous slide. And the strongest ones are typically things like aromatic functionalities binding to aromatic functionalities. And note that I'm showing this as an edge-to-face interaction. That's a very common mode for side chains to interact with each other. Another way is through a dispersive force. This was that van der Waals interaction that I introduced to you back in chapter 2. Again, these are common because they provide a lot of binding energy, and this sort of binding energy can be used to drive protein folding. Less common, and perhaps surprisingly so, are the ones that are more predictable. Okay, so you can readily predict a charge-charge interaction on, say, the surface of the protein. But it turns out these are relatively weak. And do you know why? So why are these weak interactions, charge-charge interactions?
They depend on the pH.
Depends on pH? Yes. Yes, I heard a couple people say aqueous or in water. In these aqueous or in water environments, oftentimes there's counterions which can partially shield these charged functionalities, or the water itself is competing for hydrogen bonding to these charged functionalities. And that has the effect of weakening the charge, lessening the charge, and making these charge-charge interactions much weaker than they might otherwise be expected to be. And so let me show you one example of this. This primary amine functionality, it would be found on a lysine side chain. And nearby chloride ions from sodium chloride could readily interact with this primary amine. If the chloride ion is interacting very closely, that's going to partially neutralize this positive charge. Right? Chloride is negative, the positive charge is positive. The two of these are going to be neutral. And so the net effect is that this interaction is much weaker than it would otherwise be. Over here, the hydrogen bonding ones are also unexpectedly weak. And again, that's because they're going to be competing with water. Water is the ultimate hydrogen bond donor and acceptor, and it's capable of readily forming hydrogen bonds, and this has the effect of weakening designed-in hydrogen bonds in ways that are very maddening. Okay, if you're a medicinal chemist somewhere and you design the perfect hydrogen bond, you might be shocked to find that you're only gaining, say, half a kcal per mole of binding energy, which is very aggravating because you might have done a lot of work to install that hydrogen bond. Okay, let's take a close look at two proteins binding to each other. And what I'm showing you is one half of a protein-protein interaction. At the top, this is protein 1, and in blue down here, this is protein 2. I'm coloring in the atoms according to their identity. So for example, oxygen is colored here in red, nitrogen in blue, and sulfur in yellow. Okay? Notice that the interface of the second protein, protein 2, is dominated by hydrophobic functionalities. Okay? There's a lot of carbons and hydrogens down here. There's no hydrophilic functionalities, carboxylates, hydrogen bond donating carboxamides, etc. It's almost entirely hydrophobic stuff. So at the interfaces between a receptor and its ligand, the big money in terms of the interaction energy is dominated by these aliphatic or aromatic functionalities. Okay? They're the ones that are dominating down here. Up here at the interface with water on the outside of a protein or on the outside of a protein-protein interaction, there's lots of charged functionalities, such as this carboxylate side chain from a glutamic acid, this amine functionality. There's lots of hydrophilic functionalities that can interact with the water. This has the effect of orienting the protein in this interface between one protein and water. Okay? Makes sense? This should make sense, right? We also alluded to this earlier in the quarter when we talked about the hotspot of binding energy, when we talked about non-covalent interactions. So this is kind of a concept that we've seen a couple of times now. Okay, I want to talk to you next about peptide structures and why peptides are useful, and then we'll get on to proteins. Okay, so here's a antibiotic called cyclosporin A. This is actually given to transplant patients who have had liver transplants, other types of transplants, as a way of suppressing their immune system. It's an immunosuppressant. Notably, this is kind of a big exception amongst peptides because it can be given orally. Okay, meaning that you can give it to someone in pill form and they can take it that way. I should note that in the United States, culturally, we prefer pills that are given, or we prefer to give pharmaceuticals in pill form, but that's not true in other countries. Other countries prefer things like sublingual, suppositories, etc. Okay, there's lots of ways of giving pharmaceuticals. In this country, though, we prefer to develop drugs that can be given in pill form orally. The problem, though, is that when you give someone a pill orally, it drops into their stomach, where there's all kinds of proteases to hydrolyze amide bonds. Okay, and so if you give them a peptide that's unstructured, those proteases will catalyze the hydrolysis of the amide bonds. Okay, and that in turn will result in the destruction of the pharmaceutical, making it ineffective. right? If the pharmaceutical gets digested up readily, it's not going to be useful as a drug. Maybe it's a good amino acid supplement, but it's not going to be useful as a therapeutic. And so the kind of big exception to this are peptides that are cyclized. Okay, notice that this does not have any free or N or C termini. The N and C termini of this peptide has been hidden away as the peptide has been made into a ring. Okay? It's entirely ring shaped, so it's been cyclized. Turns out this is fairly general, that you can take peptides and connect up their N and C termini if they're close to each other in space, and in doing so, make the peptide highly resistant to being chewed apart by the peptidases, by the proteases found in the stomach. Okay, this actually works pretty well. It's used very commonly in drug development. Some other little things about this that happen to make it even more resistant are, notice that each one of these nitrogens are largely, with this exception over here and this one, these are largely methylated. The little line here, that indicates a methyl functionality. That in turn also makes it very hard for the proteases to digest the amide bonds of cyclosporin A. So this is really a major exception. The other thing is that the loss of these NHs turns this amide into being something that's largely hydrophilic into something that's a little bit more hydrophobic. And this helps the peptide slip through the membranes that are found on the surface of the cell, okay? Those plasma membranes. Okay. So let's talk a little bit about what you can do with peptides. Peptide binders to particular targets, relatively easy to discover. There's many sources of these. Some sources we've talked about. You can use phage display. You can use a variant of aptamer display, a messenger RNA display. You can also search through organisms found on the planets, like this pit viper shown here, and identify venom peptides from the pit viper. So this peptide works by binding to a particular active site. Okay, so if you could solve the structure of this bound to the active site, or do a lot of structure-activity relationship studies where you mutate these side chains and identify one half of the peptide that's working, you can make an analog to the original starting peptide. Okay, let me explain that a little bit. So you have your pit viper. Somehow, and I don't know how, but you do this part very carefully, you collect the venom, okay? And then you start looking at various fractions of the venom to identify the active ingredient that's making it effective as a, I think in this case, this is a anti-inflammatory or pain-suppressing peptide, okay? So you look through that, you identify its active peptide, which happens to be this long peptide over here. You then make a bunch of versions of this long peptide that are shorter. Okay, so you make one that has the 5 amino acids at the C terminus, one that has the 5 amino acids at the N terminus, and you test each one of those. Okay, this is some work, okay, but it's not so hard. It's pretty easy to synthesize peptides. The book provides more details on that. Okay, so if you do that, you will eventually find that you don't need this big long peptide. Instead, you could get away with just this shorter analog peptide. And then if you spend a little bit more time in this, you might find some amino acid substitutions that make it more effective. So replacing, for example, this ring-like amino acid side chain proline with, in this case, alanine. Okay, so you're making it a little bit simpler. You're lowering the molecular weight. Next step here is to make analogs of this lead peptide over here and try to find a simpler variant of it. Okay, so you go down here to the simpler variant, and then this thing, it turns out, is not so orally available. But if you make this version over here, this actually is orally available as a drug. Okay, so this is actually a prescribed pharmaceutical, and this actually survives in the stomach. Okay, so even though it has an amide bond, it's not getting chewed apart by peptides, by proteases. It's surviving in the high protease-rich environment of the stomach. Okay, so all of this stuff over here, This is what I was talking about earlier today when I was talking about all the stuff that medicinal chemists are doing and then these chemical biologists and biologists. That's the stuff that we do. Okay, we're going to start with some large peptide lead, try to cut it down, and then try to take it and turn it into something that's orally available. And it's typically, this is done in teams of like 20 to 100 people working on a project like this. Okay? This is not done by one sole person who kind of makes it their mission in life to work out these details. Typically, that person would be part of a team with a computational chemist, structural biologist, chemical biologist to do the assays, medicinal chemist to synthesize these things. It's a big team. Okay, it's a big team effort to get to here. And then this compound over here might sell, hundreds of millions of dollars, if not billions of dollars a year in sales. Okay? Okay, so let's talk very briefly about the synthesis aspect of this. This is all I'm going to be showing you. After this, I would like you to review this very briefly, this topic, and then skim through the book. Okay, so here's what I would like you to know, and this is essential knowledge. What I would like you to know is that if you mix together two amino acids in a high concentration, what will happen is you'll get an exchange where the amine functionality of the one amino acid and the carboxylic acid functionality of the other amino acid will exchange protons. Okay, so the proton on the carboxylic acid will protonate this amine, giving you an ammonium ion and then also a carboxylate. Okay, so that exchange happens very, very rapidly. Okay, but no amide bond results. You can mix these two together for 1,000,000 years. Nothing will happen. Furthermore, you could heat these up for a very long time, and yeah, you'll get a little bit of amide bond coming out, but you'll largely get a lot of other junk as well. Okay, you get all kinds of side products when you start heating things up to high temperatures. Okay, so instead, what we do in the laboratory is use some sort of carboxylate activation reagent. This is analogous to the tRNA that formed that activated ester. The amino acyl tRNA that I showed last week that activates the amino acid, this is analogous to that. And the way we do this is we use an activation reagent that's typically a carbodiamide. Okay, this is an isopropyl variant of the dicyclohexyl carbodiamide that you learned about back in Chem 51C. Okay, so this is the equivalent of DCC. You remember DCC? It's like DCC. Okay, so this thing is a fantastic electrophile. It's an even better electrophile if it's protonated. Okay, this then acts as an electrophile to be attacked by the nucleophilic carboxylate to give you this key intermediate, which is a acyl isourea. Okay, this acyl isourea is now an activated carbonyl. Okay, so this is all nicely teed off to form an amide bond because you've turned this oxygen adjacent to the carbonyl into the world's greatest leaving group. Okay, key step. This means then when you add the amine, you can then readily form, you can have the amine attack this carbonyl, electrons get kicked up here, electrons come back down, kicking off a good leaving group. And that good leaving group that gets kicked off is a urea. Notice that this gives you a carbon-oxygen double bond, which as we've talked about is very strong. Okay, so this goes back to when we talked about tautomerization of the DNA bases, and I pointed out the strength of a carbon-oxygen double bond. Here we see it strike again. That strength is key to understanding how this reaction works. Okay, it also helps if in the case of DCC, the urea precipitates out of solution, driving the reaction forward by Le Chatelier's principle. Okay, in this case though, this diisopropyl urea, not so insoluble. Okay? Oh, and by the way, for the aficionados in the audience, if you're wondering why we're no longer using diisopropyl carbodiamide, oftentimes we switch to the from the dicyclohexyl to the diisopropyl because chemists that spend a lot of time doing these reactions, and believe me that you end up spending a lot of time doing this stuff. When you spend a lot of time doing this stuff, small quantities of DCC that get on in your skin turn into a potent allergen. Okay, so this will also react with carboxylic acids found on the surfaces of the proteins in your cells and on your skin, and in turn, that will lead to a massive immune reaction. So what we do is we switch around between the dicyclohexyl variant to the diisopropyl variant, and we also try to be extremely, extremely meticulous about not inhaling this stuff, about not allowing it to contact our skin, etc. That is really, really essential. This stuff is really nasty. Okay, and you know, I have friends who can't even go into certain laboratories because they're that sensitized to this type of chemical. Okay, so this is the level of detail that I would like you to know to understand peptide synthesis. The textbook provides a whole lot more level of detail, and I'd like you just to kind of skim through it. Okay, don't stress about it, just skim through it. But I'd like you to know this particular reaction. This one is useful. This is the reaction that I've done a lot, that I think almost everyone in Chem Bio has used many, many times. Okay, so after the peptide is synthesized, how are you going to get to a larger protein? Okay, two ways. Way number one is to coax bacteria cells to synthesize the large protein directly for you. Okay, we talked about that in the context, I believe this was week 1, where we talked about E. coli acting as little factories to synthesize proteins for you. Okay, so that's one way to do it. A second way is to stitch together a bunch of short peptides, and in doing this, then you get to a much longer peptide that eventually becomes its own protein. Okay, it turns out that's actually kind of non-trivial. Each one of the peptides has functionalities dangling off the side chains, et cetera. So it's not as easy to do that kind of thing as one would like. So instead, we've invented a series of reactions that allow us to do this kind of convergent synthesis to make a larger protein. And the way this works, here's one example. This is one called native chemical ligation, discovered by Steve Kent. Here's the way this works. You have a thioester at the C-terminus. Okay, so this would be the carboxylate. Instead of a hydroxide over here, there's a sulfur and then an R. R could be something like a phenyl functionality. Okay, so that's called a thioester. This happens to be even more reactive than the regular esters. Okay, and we know esters are reactive because we saw an example of that with the amino acyl tRNAs, which were esters. Right? Okay, so we start with this very reactive functionality, and this can do something called trans-esterification or thioester exchange, where this SR functionality is kicked off, this thiolate functionality is kicked off to give us a free thiol, and in turn, a cysteine side chain, the sulfur of a cysteine side chain, nucleophilically attacks this carbonyl. Why would it bother doing this? Well, part of it is driven by the tremendous nucleophilicity of sulfur. Sulfur is the king of nucleophilicity. It loves being a nucleophile. Part of that is if we look over here at the periodic table. Okay, so sulfur is one row below oxygen. These electrons are a little bit further away from the nucleus. And being a little bit further away, they can be more readily given up. They're a little bit further out of orbit, so they're more effective as a nucleophile. So again, sulfur is the champion of nucleophilicity. Going down the periodic table, selenium is even better. Tellurium oxidizes so quickly we don't even see it. Selenium, though, is even better than sulfur, but sulfur is our grand champion, at least in Chem 128. Okay, so sulfur attacks this activated thioester. That in turn gives us this intermediate, and this intermediate can then do a attack where this amine, the lone pair in this amine, attacks the carbonyl, giving you a kicking electrons up to the oxygen, which in turn collapse down, and in turn give us a new amide bond and booting off the sulfur of the cysteinyl side chain. Okay, now why is this useful? This is useful because it gives you a way of stitching together two big peptides. Okay, peptide 1 over here is indicated by this little squiggle. That could have 20 amino acids going off to the left. So imagine 20 amino acids going this way. Peptide 2, again indicated by a little squiggle, 20 amino acids going this way. And here's the thing. If you mix these two together, you will get an amide bond out of it uncontrolled, without even thinking about it. All you have to do is mix the two together, maybe control the pH with a little pH buffer, and boom, you get amide bonds out of it. It works that well. Okay, so that gives us a way then of taking 21 over here, 21 over here, put them together, and you get 42. If you do that a couple more times, you're talking about really big assemblies of proteins. This is how you could synthesize really big structures that start to have some real consequence for your experiments. Okay, so this works really well. It's used very commonly. It's often used to incorporate unnatural amino acids, a little bit different than the suppression technology that we talked about in the context of RNA. But in this case, you could just chemically synthesize whatever you want, including unnatural amino acids, including weird backbones that I'm not talking about today. Okay? Chemistry this elegant must have a natural analog. And it turns out that when we look closely at cells, we find that cells can also do a self-splicing reaction, where proteins newly synthesized by the ribosomes will actually cut and paste themselves apart without any intervention from other proteins. Okay, so there's no other catalysis that's available. Here's the way this works. So some protein's been synthesized by the ribosome. Here's the N-terminus, here's the C-terminus. And by a mechanism that I'll show on the next slide called protein splicing, an intermediate between two exteins is cut out, is actually physically removed. This is analogous to the introns of the messenger RNA. Right? We talked about splicing a messenger RNA. Here we're seeing something analogous to this, but instead of having the RNA backbone, this has the protein backbone. And again, this also is something that happens spontaneously on its own. You synthesize this thing, you put it in water, and before you know it, the intein has dropped out. Why don't we take a look? The mechanism recalls what I showed on the previous slide that we called native chemical ligation. It involves a similar rearrangement of the backbone of the protein. Okay, so let's see. Protein splicing to remove inteins. Here's the way this works. Flanking on the N-terminus of the intein, there's always a cysteine functionality. That gives us the wonderful nucleophile of the thiolate functionality of cysteine. Okay, so this thiolate can now attack the backbone of the extene. Okay, when that happens, you get this intermediate structure, and then by an N to S acyl transfer, this other flanking cysteine can attack this thioester. Okay, so this is kind of like, I don't know, it kind of reminds me of like a trapeze artist. Okay, we're going to basically be passing off an activated carbonyl. We're passing it off from one person to another, etc. Okay, so high wire act. Okay, so this is the thioester exchange. This is analogous to the first step of the native chemical ligation that I showed you, a very analogous thioester exchange reaction. Okay, so again, thioester exchange over here, that has the effect of bringing these two exteins next to each other, and in the final step, asparagine functionality, the side chain of an asparagine functionality, attacks the carbonyl backbone, freeing up the intein down here and giving you this imide functionality. Okay, and notice now that the backbone has been restored, the two exteins are bound to each other covalently through amide bonds. This works really well. Okay, and it's actually found naturally occurring in something like 300 different proteins in various organisms. In fact, New England Biolabs has a whole website that's dedicated to tracking these. So if you're interested, you can look this up on Google and you can find a big listing and all kinds of examples. In order for this to work, however, there is conserved sequence. This isn't going to work with just any protein. Okay, this really requires, for example, the cysteine at the N-terminus of the intine and another cysteine at the C-terminus of the intine, and then a key is this asparagine right here. Furthermore, the intine has to bring the two ends kind of close to each other, right? If the intine is set up so the two ends can't find each other, then structurally this cannot happen, right? If the two ends are not, wandering around nearby, no reaction is possible. So for these reasons, we find lots of conserved sequences within the inteins, and certainly all the amino acids that I've depicted on this slide, because that's its mechanism. Okay, makes sense? Questions about anything we've seen so far? Okay. In that case, let's talk next about conformational analysis. When we look at proteins, we find that they're not just flopping all over the place. Amide bonds, for example, which hold together proteins, occupy only a small amount of three-dimensional space. That there's really limits on rotational angles within the protein. And this topic we're going to call conformational analysis, and we're going to look at it in some detail over the next 15 minutes, and then the next lecture we'll cover it in much greater detail. Okay? So that's our plan. I want to talk to you about how to look at structures and be able to predict their predominant conformation. I'm not going to call it the one and only one conformation, because in fact what we find is kind of a bell curve of conformations. This is shown by this upper graph over here. So this is a histogram, meaning that higher numbers over here, higher on this y-axis over here, or z-axis over here, means more representation. And notice that there is a predominant peak, but there is some population that exists in these otherwise flawed rotation angles, okay, where I've defined some rotation angles down here. So yeah, there's going to be some dominance of one particular conformation, but for the most part, there's also some other rotations that are possible. The goal, again, though, is for you to be able to predict what that dominant conformation is going to be. And it turns out that actually just using exactly what you learned back in sophomore organic chemistry, you can make predictions about what the amino acid side chains are going to look like. And then in turn, we can start to understand how proteins can fold into particular conformations. Okay, that's our goal. Let's talk first about the hydrogen bond. Okay, let's start basic. It turns out that hydrogen bonds are not flying all over the place. And this is set by the fact that the hydrogen bond acceptor are the lone pairs on oxygen, which look like these Mickey Mouse ears. Okay, so here's the oxygen, here's the orbitals that encompass these two lone pairs. Notice that they're not going all over the place. They're like Mickey Mouse ears. They have only one particular orbitals that are two particular orbitals that are involved up here. The electrons are kind of confined to those orbitals. They're not going all over the place. And so for this reason, when we look at hydrogen bonds to this oxygen, we find that the hydrogen, oftentimes, it can be found at an angle that sets it up to be occupying these orbitals or to overlap with these orbitals. Okay? So yeah, we've talked about this before, but the restrictions inherent to hydrogen bonding limit the flexibility of the protein backbone. Turns out that the flexibility of the protein backbone is largely dictated by hydrogen bonding, by the amide functionalities of the backbone. Let me show you an example of that. Okay, so this is just a backbone. I've removed all of the amino acid side chains, and in dashed lines, these are the hydrogen bonds. Notice that many of these hydrogen bonds are coming off the oxygen at an angle. That's to take advantage of the Mickey Mouse ear shape to the lone pairs on the oxygen. It's not totally perfect, right? This hardly looks Mickey Mouse shaped, but you know, they kind of look Mickey Mouse shaped. This one looks a little straighter. Some of these look a little more Mickey Mouse shaped than others. Okay, so again, you know, this is more of a guideline, and there's considerable other angles that are available as well. Okay, these are not definite, you know, set in stone kind of things. These are more like preferences. Okay, so this is the structure of a protein, a beta sheet protein called concavalin A. I'll talk more about beta sheets in a moment. But it demonstrates the variation in the geometry of hydrogen bonds that's possible, all the way from straight to angled. But in general, angled predominates. Okay, and notice that the hydrogen bonds we're talking about are between the NHs of an amide from the backbone to the carbonyl of a backbone, where again, the carbonyl is the hydrogen bond acceptor, the NH is the hydrogen bond donor. Exactly what we've seen before. This kind of, to me, looks like a zipper, right? So this is kind of like the zippers enclose. And so these hydrogen bonds are relatively weak, but when you get enough of them together, you get a very, very strong interaction, like a zipper, right? You know, each one of those little hooks in the zipper, not so strong, but you zip the thing up and you get enough strength out of it. Okay, so that's a very similar concept to what we're seeing here. Okay, each one of these, maybe half to 1 kcal per mole, nothing to write home about. You put a bunch together though, now you're talking about big money. Okay, now another canonical mode for protein backbones is to form helices. So we saw on the previous slide that the backbones can form sheets. These sheets are typically curvy. We'll talk more about that in a moment. In addition, the hydrogen bonding preferences can force the backbone of the protein into a curvy helix that looks like this. If the helix has a pitch such that the I residue is interacting with the fourth residue down away from it. Okay, so if we count, actually let's start at the top. Okay, so over here, if the NH of an amino acid is forming a hydrogen bond with its own carbonyl, that would give us this weird structure over here. Okay, so in pink, these dashed lines indicate the hydrogen bonding. We don't see this. Okay, so this would be I to I interactions. We don't see this at all in protein structures. Okay, why? Well, this kind of cakes the whole thing into a conformation that's not so comfortable, and hydrogen bonds down here can better satisfy the requirement for overlapping with those lone pairs on the oxygen. Okay, notice over here the lone pairs on the oxygen, not so Mickey Mouse-eared, more like coming off on the sides. Okay, so if we interact with the next amino acid, the carbonyl interacting with the I+1 amino acid, that gets us, again, a kind of twisty looking structure, not so strong. Notice now that the hydrogen bonds are at terrible angles. Okay, oh, something else I should have pointed out. I didn't. Notice that the NHs are pointing directly at the carbonyls, again, within those angles. That angle really matters a great deal. When the angle is broken, that sets up a much weaker hydrogen bond. And I know that's something we've talked about, but I wanted to remind you. Over here, weak hydrogen bonds, right? The angles here set up a very weak hydrogen bond. The NH is going off in space one way. The oxygen carbonyl is over here, does not set up a good hydrogen bond. This is too angled. Similarly, over here, too angled. Where things start to get interesting is when we get to interactions between a carbonyl on I residue and an NH on I+3. So that's 3 residues away. That sets up a hydrogen bond that's nearly the right angle. And this kind of very twisty helix is called the 310 helix. It's observed. We could find 310 helices. They're important in cell signaling for binding to, for example, SH3 domains. Okay, so we can find those, and we'll talk more about those later. And the reason we could find these is that the hydrogen bonds for the 310 helix, not so bad actually, right? They're not so bad because actually the NH is kind of pointed in the right direction. There's a kind of a nice the oxygen, the lone pair on the oxygen gets to stay in its Mickey Mouse orbital. This is not so bad. Okay, so those actually do happen. So these 310 helices are a very twisty helix. This is a really tightly wound, a very tightly wound helix. A more common helix is called an alpha helix, and it consists of an interaction between a carbonyl on the I residue and the NH of an I+4. And this gives you hydrogen bonds that have nearly perfect geometry, nice and straight with each other. And so these alpha helices are very commonly found. It's an important motif of protein secondary structure. Very, very common. Okay? Yeah, question over here.
I just wonder if there is any time there's a mixture of the interaction, or it's always like I+4.
Yeah, okay, that's a great question. What is your name again?
Oh, Ashley.
Ashley. Okay, so Ashley's question is it always one thing or the other? Do you sometimes see the two things interconverting? And the answer is we largely see dominance of these alpha helices. And when we look at 310 helices, oftentimes we'll see some flickers of going to I+4 and then going back to I+3 and I+4 and I+3. And so, yeah, there can be a mixture of the two. And there's techniques that you can use, like circular dichroism, to follow those two and separate out what percent is 310, what percent is alpha helical. So thanks for asking. Okay, our goal here is to maximize the number and the quality of hydrogen bonds between backbone atoms. If we do that, we're going to have a much more stable secondary structure. Okay, so that's kind of the ultimate goal for everything that I'm talking to you about today. Let's talk next about alpha helices in general. Alpha helices often clump together with other alpha helices into large assemblies. And it turns out that the alpha helix itself has a dipole associated with it. Okay, and you can see this up here. Notice that all of the carbonyls are pointing in the same direction. Each one of these carbonyls has a tiny little dipole associated with it. We talked about dipoles when we talked about dipole-dipole interactions much earlier in the quarter. At the time, I told you that dipoles were separations of charge, where in this case, the oxygen, being a little more electronegative, has a little bit more negative up here and a little bit more positive at the carbon of the carbonyl. And so this little dipole over here, this little dipole, all these little dipoles are all pointing in the same direction. So if you sum up all those little tiny dipoles, you end up with a much larger net dipole. So the helix itself has a fairly large net dipole. And that means that one end has a negative charge and the other end has a positive charge. And I would tell you that when I first learned about this, it was I thought of it as kind of a myth. I've come around entirely. And I can show you the best example of a helix dipole in action. This is the outer surface of the filamentous bacteriophage that's used for phage display. It consists of a series of alpha helices. Okay, so it's like these 50 amino acid long alpha helices that are coiled around each other. They're actually coiled in an unusual arrangement. And this is a cross-section through the virus. And you can see all the alpha helices stacking on each other. In the very center down here is the DNA of the virus. Okay, and it's not shown. I've left it out to make this simpler. Okay, but notice that these actually form a right-handed coiled coil. Okay, unfortunately, I hate to do this, but I'm starting with the major exception to all coil coils. Every other coiled coil that we're going to see this quarter, and in fact, 99.999% of all coiled coils found in nature are left-handed coiled coils. This one just happens to be right-handed. But here's the thing. Because each one of these alpha helices has a dipole, and all of these coils are pointing in this, all these alpha helices are all pointing in the same direction, the virus ends up with a massive net positive charge up here, net negative charge down here. And the effect is that the virus itself can be oriented in a magnetic field. Okay, it has enough of a dipole that if you put it in an NMR tube, it goes boing and orients in the magnetic field of the NMR tube. And to me, that's pretty persuasive evidence that actually this alpha helix stuff might have a dipole associated with it. Furthermore, when we look at lots of protein structures, we find that the positive end of this alpha helix dipole is oftentimes pointed at the negative charge of a functionality in the protein. And I'll show you an example of this. Okay, so here is a sulfate atom and a sulfate binding protein. And look at these alpha helices that are pointing directly at the sulfate. That extra little bit of positive charge could come in and stabilize that negative charge. We also find this when we look very closely at enzymes that have to do things like transfer phosphate functionalities, which also have lots of negative charge. Okay, let's stop here. When we come back next time, we'll be talking about protein structure and my favorite topics.